Thanks, Ryan, and thanks, Brian. That was off to a great start for our 10-year anniversary. I guess I just want to start with saying happy birthday to DCMB. So we had a recent lab meeting. Not everyone was there, but a lot of folks were. So from top left to right, from Keegan, myself, Maya, Alice, Loveland, Peter, Cassie, Arushi, Krista, Vlad, Adelaide, and Cynthia, happy birthday. I've started here in 2014, so I've been here for 8 of those 10 years, and it's been an absolutely phenomenal experience. Like Brian said, the environment is top notch, from undergrads to graduate students to postdocs to staff to administrators and to my faculty colleagues. It's really a privilege to be up here and share this platform with so many impressive individuals. So happy birthday, DCMB. I want to talk a little bit about one of the projects that we're pushing on nowadays. And here's the kind of outline of the brief talk that I'll give. First, I'll start with some motivation about biological complexity across dimensions, and the need to do data integration is absolutely critical to understand mechanisms across these dimensions. Then I'll talk about, really the meat of the talk is population scale, integrative single nucleus multi-omics to dissect GWAS signals. And I'll focus on T2D and related traits and skeletal muscle. Have some conclusions, and then I'll delay questions until the very end after everyone has had a chance to speak, and we'll all have a seat up here. So T2D is quite a complex disease. It's characterized by glucose dysregulation, and its effects are manifest through multiple different organ systems: the brain, the liver, the pancreas, the islets in the pancreas, skeletal muscle, and adipose tissue. To some extent, we're involved in analysis of all these tissues. Today, I'm going to focus on skeletal muscle tissue. If you look at T2D from a different perspective, from genome-wide association study or GWAS perspective, it's an incredibly complex disease, right? So currently, the most recent high-powered GWAS has over 600 statistically independent signals of genetic association across your genome. So your genome is literally littered with risk alleles scattered everywhere. And if you look closer at these risk loci and figure out where the SNPs occur, it's quite remarkable in that the vast majority of them are in the non-coding space, right? So that little bar plot in the light gray is all the non-coding signals, and the dark gray is the coding, and it's less than 10% of these signals overlap a coding feature. That means that really what's happening here is, in purple is a regulatory element, and the green triangles are T2D and SNPs overlapping that distal regulatory element that loops and contacts some target gene and then modulates expression of that target gene. So most of these variants that are associated with disease are not directly altering protein function, but instead modulating when, where, and how much genes are expressed. Thus, the causal mechanisms at any given locus is incredibly difficult to dissect. And I want to back up a little bit and say that T2D is not the only disease or trait for which GWAS has been performed. If you look at a recent release of the GWAS catalog, there are over 72,000 documented genetic associations with some sort of disease or a quantitative trait. And if you look at all of those associations throughout the GWAS catalog, it's exactly the same picture as T2D. So they're nearly all non-coding. So whenever I talk about T2D, or I'll talk about BMI or waist to hip ratio or any kind of complex trait, just recall that these are models for almost every complex trait in which nearly all SNPs are non-coding and not affecting directly genes. So the way that we think about this kind of hierarchy of how do genetic variations exert this complex trait effect on an organism, they percolate through these layers of molecular hierarchy. And I've arranged them here to show the epigenome, the transcriptome, the proteome, all the other omes that you can think of. And this goes back to the integrative approach that Brian established when he founded the department. I'd like to emphasize that the bottom layer, the genome layer, is invariant. We're not talking about cancer or somatic variation here. We're talking about germline genetic variation, where essentially every cell in your body has the exact same instruction booklet. All the other ome layers are very context specific, right? So is it in your muscle? Is it in your adipose? Is it in your eyelid? Is it in your liver, in your brain? Did you just eat a Big Mac or a banana? Those are the types of contexts that we're talking about. I like to put a gold star next to the epigenome because I think it is the most proximal molecular layer after genetic variation that propagates the effect of a genetic variant through these different omic layers into the organismal phenotype. So it's critical to look at the epigenome, but also to integrate across genetic, epigenome, and transcriptome layers. So how do we measure the epigenome? One approach that we love in our group is called the assay for transposase accessible chromatin, or TAC-seq. And TAC-seq essentially maps the open chromatin accessibility landscape. So if you think back about how DNA is organized inside your nucleus, it's wrapped around nucleosomes right here. And then when a transcription factor binds, it focally creates open chromatin organization. And you can measure the accessibility of chromatin using enzymes like TN5. Sequence the reads, map the reads back to the genome. And at the very bottom, I'm showing you real, true TAC-seq data where to the left and right of the plot, there's basically no reads mapping indicating DNA that's occluded by nucleosomal architecture, but in the middle is highly open and accessible DNA indicating regulatory element activity. A transcription factor has likely landed there and created focal open chromatin architecture. What we're really interested in is not just any open chromatin region, but an open chromatin region that's influenced by genetic variation. For example, right here, again, real TAC-seq data, where I'm plotting individuals by their genetic variation at this particular SNP. So individuals that are homozygous A have very high chromatin accessibility, individuals that are heterozygous have medium chromatin accessibility, and individuals that are homozygous G, you basically wouldn't call a peak there at all. It looks completely flat, right? So a very strong genetic effect on the regulatory landscape, which we can quantify by doing this TAC-seq readout. So this is what we want to get to when we want to do this at population scale, not just using reference maps, because again, if your reference map is based on an individual with homozygous G genotype, you've totally missed the point here. So focusing on skeletal muscle, it's a bulk complex tissue, which requires a single nucleus or single cell approach to dissect all the heterogeneity embedded in that complex tissue. And on the right are the types of cells that you would expect to see if you were to do single nucleus or single cell profiling of skeletal muscle. The slow and fast twitch muscle fibers are multinucleated, so you'll expect to see lots of nuclei from those particular cell types. And then you also expect to see mesenchymal stem cells and immune cells and cells that represent the vasculature, like endothelial and smooth muscle cells, satellite cells, the precursors, and NMJs or neuromuscular junction cells that help with communication between the skeletal muscle and the nervous system. So I'll just highlight that back at the end of 2021, we did work in collaboration with Chuck Morant here and co-led by three co-first authors, Peter Orchard, Nandini Manikam, and Krista Ventresca, where we did single nucleus multi-omic profiling across human and rat, both RNA and both attack modalities. This represents about 34,000 nuclei. So I'm not going to talk any more about this other than say it exists. And at the end of 2021, fast forward a few months, the human Tabula Sapiens Atlas came out and made the cover of Science. They had skeletal muscle samples in there too. It was RNA data, not attack data, and it represents about 30,000 different nuclei. So a little bit lower than we had published a few months earlier, but a very impressive paper showing in total nearly 500,000 nuclei, about 483,000 nuclei, but across only a few individuals, not a lot of individuals, just like 4 or 5 or less than a dozen individuals. So what we've done, and this is all in published work, is single nucleus profiling across over 400 different samples, and collectively this represents a little over 500,000 nuclei. And we've profiled these in samples at 3 different levels: at the genetic level, so the genome, at the single nucleus chromatin accessibility level of TAC-seq, and also at the single nucleus transcriptome level. I'm going to fast forward through a lot of the QC and details, but generally show you what our aim is to identify nuclei that correspond to the different fiber types, to the endothelial cells, the satellite cells, all the constituent cell types, and then integrate those cell-specific transcriptional and chromatin accessibility profiles with underlying genetic variation to identify on the top here genetic variants that are associated with chromatin accessibility, or a CA-QTL, chromatin accessibility quantitative trait locus, or genetic variants that are associated with gene expression, or expression QTL, eQTL. And this, again, will illuminate kind of how genetic variations influence the transcriptome and epigenome, and if one of them might have a cause on the other. The way that we did this experiment, I thought, was pretty fun because it's scary up front. We took these over 400 individuals, and we only used 10 batches, and each batch had about 40 individuals mixed together in the batch. So every of those 40 samples was processed in exactly the same manner. And the way that we can get unstuck and assign a nucleus back to the individual that it belongs to is because all of these samples were previously genotyped. So we just take the RNA-seq reads or the attack-seq reads, and we compare it to the known genetic variation of that individual, and then we can assign an individual nucleus from one of the 40 samples to its corresponding donor. Pulverize, mix everything up. Again, everything very well controlled in these batches, and we implemented a randomized block study design to control for any kind of covariates of interest. We did lots of attack and RNA single nucleus sequencing, and the value of this is it's super cost efficient because you can massively overload these flow cells when you're doing this type of multiplexing, because you're not scared of doublets anymore. You can genetically identify doublets and not be worried about them in your analysis. That's point number two. You avoid batch effects, right? So all these people from different ages and different disease statuses and different sites across the country are all mixed in the same batch. So it eliminates any kind of technical variations associated with processing samples individually. It's incredibly fast. We did all of this data generation in about 1 month. So you would not be able to do that unless you had an army of personnel, which we don't, working on single samples around the clock. And then finally, I think it increases rigor and reproducibility of the work because, again, it very nicely controls for technical and batch effects in the data, of which there are many. So I'm going to fast forward to the analysis that we got. This is only looking at a subset, 287 of the 400-ish samples. You can see we get different fiber types, type 1, type 2A, type 2X, and then a bunch of other different cell types that we expect, all at low abundance. In the bar plot, you can see we've quantified, based on both modalities, the attack on the left and the RNA on the right, how much of the different fiber types we observe, quite a bit, all the way down to adipocytes and less than 1% of the data in your skeletal muscle. It's kind of cool. So now we focused on the most abundant cell types for which we would have the greatest statistical power. And we did our integration with genetic variation data. So cell type specific and type 1 fiber, type 2A fiber, type 2X fiber, mesenchymal stem cells, and endothelial cells call eQTL and CA-QTL. And we get a lot of these, right? And so the table here represents the different cell types on the y-axis and the different categories, either eQTL in blue, and you can see we're getting a few thousands of these, and it's based on the abundance of the cell type. So the more of the nuclei that we get, the better power we have to detect QTL. But the CA-QTL, we get orders of magnitude more, right? So there's much larger, I think, effects, genetic effects on chromatin accessibility than on gene expression. So the next thing we wanted to do is go back and compare to some of these atlases that are widely used in the field. So there's a Cell paper that was recently published, a single cell atlas of chromatin accessibility in the human genome, and they have skeletal muscle here. And I know you probably can't read that, but there's about 40,000 nuclei from skeletal muscle where they did attack-seq. So we wanted to look at our CA-QTL, especially where there's a genetic effect, and say, is that CA-QTL the most important kind of chromatin peaks in the genome where genetic variation affects it? Are they represented in the cell atlas? And again, here's the example. What if your atlas had an individual with this genotype GG instead of a heterozygous AG or homozygous A individual? And we reasoned that the presence in the atlas would be based on two things at the CA-QTL peak. One, the effect size. The genetic variant have a big effect. This has a whopper effect, right? Like if you have the wrong variant, you're basically flat and you wouldn't call a peak there. Also the minor allele frequency. What's the frequency of the allele that corresponds to open accessible data? So here's what happens when we compare our CA-QTL data to the published reference atlas. On the x-axis here is the effect size of the genetic variant. On the left, this is a variant with a small effect. On the right, This is a variant with a very large effect. And the y-axis is binned by minor allele frequency of the allele that corresponds to open accessible chromatin. And you can see rare alleles that influence chromatin accessibility at the top, and these are very common open accessibility alleles at the bottom. And if you're down here in the part of the graph, these are common alleles that affect chromatin opening, and they have big effects. Basically, every single one of those, 100% of those peaks are detected as expected in the reference atlas. But as you move to smaller effects and rare alleles, you're missing. Corresponds to about 30% of the peaks are in the atlas. So you're missing about 70% of the most important peaks in the atlas, which we think is quite important going to like population scale. All right, so I will, I was going to talk about some co-localization results, but I think I'm going to skip that because I'm at the zero time point right now. And I think I just included a few too many slides. And I want to say thanks again to a really diverse team. There's 6 people from the lab that are presenting posters, so go check out everything that they're doing. I think there's a lot of cool stuff happening beyond the small vignette that I was able to describe. And I'll end with saying, again, happy birthday to DCMB, and we're almost teenagers, so look out, world.